Hello students, I am Geeta, botany faculty from Sri Chaitanya Gosala campus, and you have learnt already the cell cycle topics like mitosis and meiosis. Now today we are going to discuss a particular topic, very important topic in molecular basis of inheritance in the second year, and this is very important for the NEET point of view also, and you will get many questions which are linked with this topic. And this topic as itself, it has more weightage, and even the chapter also has more weightage in the NEET. So you have to learn this thoroughly, the topic of molecular basis of inheritance, that is replication of DNA. You can see here, this is the replication of DNA. How does the DNA present in all the cells will be replicating during cell division? If you take the prokaryotes, prokaryotes will, like bacteria and all, they will undergo the DNA replication, and they have only one chromosome will be there, that is having the circular DNA, that also will undergo the replication. And in eukaryotes also, this DNA will be replicating in a particular stage of the cell cycle, that is S phase. So the replication of DNA in eukaryotes occurs in the S phase. But this S phase is a subphase of the interphase in the cell cycle. So the replication of DNA in eukaryotes occurs in the S phase of the interphase of cell cycle. Whereas in prokaryotes, the type will not be there, but generally S phase. Okay. So in eukaryotes, they have given in NCERT very clearly that in S phase of the eukaryotes, the DNA replication will happen. And what type of replication will be happening in these eukaryotes or prokaryotes, whatever the DNA containing organisms? Because DNA is the genetic material in all the kingdoms starting from Monera to Animalia. Monera, Protista, fungi, plantae, Animalia, in all these living organisms, prokaryotes as well as eukaryotes, the genetic material is DNA. And particularly it is most of the organisms will have the double stranded DNA. Actually from prokaryotes to eukaryotes, it is double stranded DNA, that is the genetic material. When it comes to the viruses, they may have DNA or they may have RNA as the genetic material. That is a separate topic, but here we are going to see only the replication of DNA, which is of semi-conservative type. Do you remember that who discovered the structure, double helical structure of DNA? It is Watson and Crick. They are Watson and Crick. So that Watson and Crick, basically the scientists who told the replication of DNA is semi-conservative type were Watson and Crick. The same two scientists who gave the double helical structure of DNA or double helical model of DNA, they only suggested a particular type of replication is happening in the DNA. The name of that replication is semi-conservative replication of DNA. According to their, when they gave the double helical model only, they said clearly that semi-conservative replication of DNA is going to happen. And in your NCERT, one quotation is there which were given by Watson and Crick in the year 1953. They said that the semi-conservative replication of DNA is going to happen in the DNA. Now you can see here, this is how the semi-conservative replication of DNA occurs. One parental DNA strand is there, this is the parental DNA, and these two are the daughter DNAs. So Watson and Crick model for semi-conservative DNA replication. The red ones are parental DNA strands and the blue ones are purple ones, okay, other color ones. These are the newly synthesized strands. So once the newly synthesized strands are created, okay, like this, now on one parental DNA, two daughter DNAs are formed in which one strand is a parental strand, red one is a parental strand only, parental strand, and the blue one is newly synthesized strand. So in the daughter DNAs, one will be parental strand, the other one will be the newly synthesized strand. So this kind of replication where in the daughter DNAs, one will be the parental strand, the other one is a newly synthesized strand. This kind of replication is called as semi-conservative replication of DNA. And they said by giving this a statement. Watson and Crick, these two scientists, they quoted this. In the year 1953, to quote their original statement that it is as follows. This is exactly the NCERT statement I have taken. It has not escaped our notice that the specific pairing we have postulated, that means the DNA structure which is having two double helical strands, that specific pairing we have postulated immediately suggests a possible copying mechanism. How the copying will be happening for the genetic material. That means how that DNA replication is happening also is included in their model. So they came to know that actually one copying mechanism is also happening in the DNA, and that is a semi-conservative replication of DNA, where the daughter DNAs will be having one parental strand and one newly synthesized strand. Okay. Now this is the experimental proof which was given. Experimental proof that was given by Messelson and Stahl. Messelson and Stahl in the year 1958 to show that it is semi-conservative replication of DNA that is happening here. So if you see here, actually they have taken the E. coli cells for the first time. E. coli, Escherichia coli cells for their experiment, Messelson and Stahl. And they worked first on the E. coli cells and they continued later in the plants, animals like human cells and all. They proved that the replication of DNA is always semi-conservative. First they started in the E. coli cells. They proved first in the E. coli cells, later they proceeded to the plants and animals, for example humans. So their experiment started with E. coli cells, and normal E. coli cells will be having N14. Normal nitrogen is called as N14. And they took that cells and they have grown actually in the N15 containing medium. All of you can see here, this red color one is the N15 containing conical flask. So they have kept that E. coli cells. This is the E. coli cell. They have kept these E. coli cells in the N15 containing medium, and this medium is ammonium chloride, NH4Cl. So they took in that conical flask ammonium chloride, and in that ammonium chloride the nitrogen what they have taken was N15. That is why this conical flask is showing N15. So ammonium chloride with N15 was taken in this conical flask. N15, all of you remember, it is not a radioactive isotope. It is just a heavy isotope of nitrogen. It is non-radioactive heavy isotope of nitrogen, that is N15. It is used in which form? NH4Cl, ammonium chloride form. And now this E. coli is kept in this flask to grow for many generations. And they collected the E. coli cell which is having wherever nitrogen is there, it has the N15. You know, in the DNA, nitrogen bases are having the nitrogen. So wherever in the cell of E. coli nitrogen is there, the normal nitrogen got replaced by the non-radioactive heavy isotope of nitrogen, that is N15. So now these E. coli cells are totally labeled with N15. So now by this experiment, first step, they got the E. coli cells with N15. All the E. coli cell wherever the DNA is there, they are having the N15. They got this E. coli with N15, and now they transferred these E. coli cells. Further they continued their experiment by transferring these cells into the medium containing N14. Now they transferred this cell, N15-1, into N14 containing ammonium chloride. So they transferred this one here and they started growing this E. coli, which is having N15 in the N14 containing medium. So the medium maintained from here onwards the N14 medium only, that is ammonium chloride with N14, normal nitrogen. So it is not radioactive, it is non-radioactive, it is not an isotope, it is just the normal nitrogen. So in that they started growing these E. coli cells. And how we can differentiate that N14 is the normal one, how N15 is the heavy isotope of nitrogen means, if you take the N15 and if you centrifuge that N15 in the test tubes, the density of this N15, it was showing near to the bottom of the test tube, almost near to the bottom of the test tube here. A particular color band will be shown with that red color. Okay, here in this I have taken the red color, so that red color is shown here. So this N15 will be collected almost near to the base of the test tube after the centrifugation. You can see that ultra centrifuge is there and density gradient. For this centrifugation, they will be using the cesium chloride. So if you want to identify how much is the density of N15 and how much is the density of N14 in any cell, you have to use the medium, that is cesium chloride. That is called cesium chloride density gradient. That you will be subjecting to the cells, and that after the centrifugation in that cesium chloride density gradient, after centrifugation the N15 will be collected almost near to the base of the test tubes, whereas N14 will be collected like in this zone. In the test tube, this is the zone of N14 and this is the zone of N15. So it clearly indicates that we have N15 which is having more density, that is collected near to the bottom or base of the test tube. The N14 which is being lighter or less in density, it will be collected almost near to the top of the test tube. So by this density gradient we can easily say N15 is heavy isotope of nitrogen and N14 is the normal one which is having less density. So N14 containing DNAs are generally called as light DNAs and N15 containing are called as heavy DNAs. And if any DNA has a combination of 14 and 15, it is called as hybrid DNA. So now the E. coli cells, first they have got with the N15 labeled, they are transferred into the medium containing N14 ammonium chloride, and they started observing every generation. You know that bacteria in the suitable conditions, it will multiply for every 20 minutes by binary fission. So as you already know that the first E. coli cells, they were grown in the N15 medium, all the cells they got the N15 and they have shown here after the centrifugation in the cesium chloride gradient, they have shown that 100%, all of them they have shown the N15 containing cells with a red band near to the bottom of the test tube. Then after 20 minutes, when they transferred this E. coli into the N14 containing medium, from there onwards, after 20 minutes what they have seen, this is the first generation. So in the first generation what they observed was, there is a band which is above, slightly above than the N15 band in the test tube. This indicates that all of them they got accumulated here, all the E. coli cells with the nitrogen containing things, they got accumulated in the middle. This clearly indicates that a middle band is formed, because you know N15 is the red band and N14 is the yellow band, but it is neither looking yellow nor looking red here. So it is in between these two, and this is called as hybrid DNA. This is the hybrid, or you can call it as intermediate DNA, where you have one will be the 14 strand and one will be the 15 strand. So after first generation, 100% hybrid DNAs were there or intermediate DNAs were there. Next they further continued in the same medium, that is N14 containing ammonium chloride, and after further 20 minutes, this is the second generation. In the second generation what happened, the DNA, they have shown the bands of, one red color band is shown and then yellow color band is shown, and this red color band is not the pure red which was showing the N15 one, just it is an hybrid one only, the same color you can see. This is the hybrid. Hybrid are 50% here, or intermediate ones are 50% here, and then the light DNA, yellow DNA which does not appear here, it appeared here, and this one is the light DNA. Light DNAs are 50%. And further they continued for 60 minutes, then it became the third generation. Okay, third generation will be having 75% of N14 normal nitrogen and 25% of N15. So, this is nothing but hybrid, because it is having one strand of N14 and one strand of N15. And in the third generation, so they have got 75% of light DNA and hybrid DNA is 25%. Further continuation of another 20 minutes, then it had shown 88% of light DNA and this is 12% of hybrid DNAs. So, if you go on for many generations, nth generation or millionth generation also, never ever you will be getting the hybrid DNA numbers will never increase. For example, here always you are getting the hybrid DNAs less in percentage. But I would like to tell you one thing, after so many generations also 2 hybrid DNAs will be there. And always if you do not change the medium, if the medium is N14, then light DNA number will be increasing or percentage will be increasing. And in this never you will get N15 pure DNAs or heavy nitrogen DNAs, you will never get. Heavy nitrogen will be always 0. So, how to calculate this based on the proof, experimental proof is, this is the diagram which is given in your textbook, NCERT textbook. So, here you can see that N15 DNA is red color and the band which is shown here is the heavy nitrogen N15 band. It is near almost to the base of this and it is appearing in the red color. Then when they have transferred that to the medium containing N14, okay, then what happened was one strand, the red one is the N15 strand and the green one is the newly synthesized strand, because the medium is having the green one N14. So, in one DNA, one parental strand is a red one and the one green one is newly synthesized here. So, this is having a combination of 14 and 15. So, these are called as hybrid DNAs or intermediate DNAs. You can see here hybrid, or you can call it as intermediate DNAs. Intermediate DNAs, because it is a combination of N14 and N15. You can see clearly here it is there near to the base, almost near to the base, but here it became towards the front. The band formed became the front, it became front band. So, it is going ahead. Okay. So, the density of this one will be in between N15 and the N14. You know that N15 will be heavier in density and N14 is lighter in density. So, N14 and 15 it formed little bit in front of the N15 band and it is given in the brown color. And after another 20 minutes, then the medium is same thing, N14 containing medium. So, from the N14 containing medium, the new daughter DNAs will be synthesized and now these 2 DNA strands will be separated and one will be given to this one and newly synthesized strand will be created. And this will be coming to the one daughter DNA and newly synthesized strand is made up of N14. The same thing happens here. And after the centrifugation in cesium chloride density gradient, they have shown that the one band is showing N14-14 like blue color band near to the top of the test tube, and one band is shown in the same brown color band with N14 and N15, that is hybrid DNA, light DNA. And after another 20 minutes or after nth generation also, what you have to remember here, if you go on going for generations, this is the first generation, this is the second generation. So, if you go on for third generation, fourth generation and so on also, what you have to remember, there is a formula to identify number of DNA molecules. 2 power n is the formula to identify the number of DNA molecules. Okay, and that n will indicate number of generations. Number of generations. Okay, so let us take one generation here. One generation. So one generation means 2 power 1 is equal to 2. 2 daughter DNAs are formed, and that daughter DNAs 100% are hybrid DNAs. Hybrid DNA. Then if you take 2 generations like this is the second generation, the number will be 4. In this 4, 50% are hybrid DNAs and remaining 50% are light DNAs. So if you go on going for many generations also, always you will be getting the hybrid will be 2 DNA molecules and the number goes on increasing. Let us see the third generation. Third generation. In the third generation, what happens? 2 power 3. How many strands will be there? 8. 8 DNA molecules will be there with 16 strands. So 8 DNAs will be there. In that 8 DNAs, 2 will be hybrid. Always 2 will be hybrid. And the rest all number will be light. Okay. So this you have to remember, and never the heavy nitrogen will always be 0. 0 is heavy nitrogen. So you can go to nth generation also, and in the millionth generation also 2 will be hybrid DNA molecules and rest all will be light DNAs, and there are no heavy DNAs in this experiment unless and until you change the medium. So here the experiment was conducted by Michelson and Stahl in the year 1958, and they proved that the replication of DNA is semi-conservative, where one DNA strand is obtained from the parent and one strand is synthesized from the medium. So it constitutes one parental strand and one newly synthesized strand. So this type of DNA replication is called as semi-conservative replication of DNA, which was proved by first Michelson and Stahl. Later, Taylor and colleagues in the year same 1958, they also proved that DNA replication in chromosomes is also semi-conservative. They started using in their experiments Visia faba. The common name is faba beans. This plant roots have been used, and second material what they used was radioactive thymidine. And by using these two, they have proved that further continuation, the experimental proof was given by Taylor and colleagues, and they said that semi-conservative replication is happening in the DNA in the chromosomes also in this plant by using the radioactive thymidine. So one experimental proof was given by Michelson and Stahl in the year 1958 by using the E. coli cells and heavy nitrogen N15 and normal nitrogen N14 in ammonium chloride and cesium chloride density gradient. Here next experiments were followed by Taylor and colleagues in the same year 1958. They also proved that DNA replication is semi-conservative only by using the material. Plant material is Visia faba, which belongs to Fabaceae family. This is a link with the first year chapter, that is Fabaceae family, and they use the radioactive thymidine. You know that thymidine is there, that nucleotide, So, that nucleotide they have used here, radioactive material, and they proved that replication is semi-conservative. So, now let us go to the actual topic of semi-conservative replication. So, now let us see the semi-conservative replication of DNA procedure and what are the requirements for that. You know, first let us see the requirements. For the semi-conservative replication of DNA. So, the first requirements or machinery which are required are mainly enzymes. Different types of enzymes are required. For example, the first one, DNA dependent DNA polymerase. This is the first enzyme which is required for this semi-conservative replication. Other enzymes like helicases, we will see all of them in detail. Helicases will be required, which are also called as unwindases. Then you have gyrases, which will come under topoisomerases. Then ligases, particularly DNA ligases. Then the primase or RNA polymerase. Then single strand binding proteins. Then energy in the form of ATP. Then the last one what we need are deoxyribonucleotides. These are all the basic requirements. Most of them are enzymes only, and here these are the proteins, and energy is required in the form of ATP and deoxyribonucleotides. These are all the required enzymes for semi-conservative replication of DNA. Let us see all of them, what they do. This DNA dependent DNA polymerase, it is meant for the polymerization of the DNA. It is required for the polymerization. Polymerization means the chain elongation, linking up the monomers together and forming a polymer, that is called polymerization. So, it will be polymerizing the DNA strand based on the template DNA strand. It is dependent on the template DNA, and based on that sequence it will be synthesizing a new DNA strand by the polymerization. That is its function, and it will always do the polymerization in 5' to 3' direction. In this direction only the DNA polymerase will be doing the polymerization, 5' to 3' polarity. Next, helicases. These are also called as unwindases. They help in breaking the hydrogen bonds between two strands of DNA, and they cause separation of DNA. They separate the two DNA strands. That is helicases or unwindases function, and they break the hydrogen bonds between the base pairs in the double helical strands. Gyrases, these actually they create negative supercoiling. Once the DNA is unwinding will start at one point and it will proceed to the other end. It is not that at a time it will be separated. At one end it will start separation like that, and it will proceed to the other end. So, when it is happening, the gyrases will actually relieve the strain, and they will relieve the strain. They relieve the strain by negative supercoiling. That means when a spring is positively coiled, okay, so from the other end you can open it up. So, in that way, once this DNA unwinding is starting, at this end they will be supercoiled too much. So, that positive coiling will be removed by the gyrases by imposing the negative supercoiling. From the opposite end it will open it up. So, in that way it relieves the strain. But this gyrase belongs to the same class of enzymes like topoisomerases. Topoisomerases, their main function, they release the tension. These are meant for releasing the tension. Releases the tension. That is a function of topoisomerases. Because once the opening of the DNA is happening, the strain is removed by the gyrase, and topoisomerases will release the tension in the open DNA, and later the single strand binding proteins will be attached to the separated strands and they will maintain the stability of the separated DNA strands. After that, the ligases function. Ligases, DNA ligases, they will synthesize some particular phosphodiester bonds between two DNA fragments. They will link up the two DNA fragments by the phosphodiester bonds. So, they are called as ligases or synthetases. Then primase, it is also called as RNA polymerase. This is a particular enzyme which will synthesize a starting point of the new DNA strand, that is RNA primer. You know, RNA primers will be there. That RNA primer synthesis will be happening by the primase enzyme. Then you know energy is required in terms of ATP. Then you have the deoxyribonucleotides. They are present in the nucleus, nucleoplasm particularly, or cytoplasm of prokaryotes, and they are the substrates. They serve for two purposes, dual purposes. They serve as not only substrates, but they provide even energy. So, these are the basic requirements of the semi-conservative replication of DNA. The main ones are enzymes, most of them are enzymes only, but some are proteins, some are energy containing compounds, and some are the substrates which are used to polymerize the DNA. These are the basic requirements. And now let us see the replication. So, in this semi-conservative replication, as I have told you already, okay, these are the helicases will be there, one type of enzyme helicase, you can see here. This will come and it will bind to the DNA strand and it will break the hydrogen bonds between the DNA strands, and it will cause the unwinding of the DNA. And that unwinding of the DNA, it will start at a particular small opening, that is called as replication fork. But that small opening will be there in a particular site, that is called as origin of replication, ORI. It is also called as origin of replication. So, helicases will identify the ORI and they will open up a small opening, they will create. That opening is called as replication fork. So, once they open it up, it will cause the twinning of the rest all part of the DNA. That tension which is created due to the opening here at the middle point or somewhere, wherever ORI is there, that tension will be released by the topoisomerases. And gyrases, they will create the negative supercoiling. So, you can imagine that helicases are going like this, and here this is the midpoint. So, you can see in one direction, the opposite direction also it is happening. Because of this, the semi-conservative DNA replication is bidirectional. This replication is bidirectional. It happens in both the directions. Okay. If you take the half also, it is going in two directions, and if you take this DNA, total DNA also, it is happening in the two directions. So, bidirectional replication of DNA is happening. And in this, first ORI will be identified, and then helicase will go and it opens up to form the replication fork, or it is also called as Y fork. It is also called Y fork. Okay. Y fork it is called as. And after separation, the single strand binding proteins will come and they will bind to the separated strands and they will stabilize the strands. And after that, the DNA polymerase, this is the DNA polymerase, like for example, different polymerases will be there. They have given the polymerases names, but one DNA dependent DNA polymerase will be there, and it will always polymerize the DNA strand in 5 prime to 3 prime direction. So, if you see here, this strand, template strand is having 5 prime, and this end it will be the 3 prime end. And for this strand it is starting with the 3 prime, and this end it will be 5 prime end after opening up. The polarity is like this. So, after opening up it is forming an Y fork, and both the strands of DNA will act as template. And one strand will be having the polarity of 5 prime to 3 prime, and other strand will be having the polarity of 3 prime to 5 prime. And you can see that always the strand which is going in the 5 prime to 3 prime, this is the newly synthesized strand which will be there, and the opposite strand will be having the 5 prime here. For the 3 prime of the parental strand, the new strand will be having the 5 prime, anti-parallel nature. So, the new strand will be synthesized on the template of 3 prime to 5 prime. This initiation point will be the RNA primer. This red one what you see, it is nothing but the RNA primer which is synthesized by the primase. That will be starting, it is the initiation of the replication of DNA. That will serve as a starting point of the new strand synthesis. So, RNA primer will be synthesized, and it will be placed exactly after the opening of the DNA. At the 3 prime end, the RNA primer will be placed. And imagine that here also RNA primer is placed on the opposite side. It will function as a 5 prime end. It will function as a 5 prime end. So, after that what happens, the DNA polymerase 3, it will bring that deoxyribonucleotides. That deoxyribonucleotides will be there in the form of deoxyribonucleoside triphosphates. They will be in this form: DATP, DGTP, DCTP, DTTP. They will have the deoxyribose sugar and adenine base, and with 3 phosphates. So, 3 phosphates will be there here. And now the last 2 phosphates are having high energy bonds, and this deoxyribonucleoside triphosphate, they will remove the last 2 phosphates. 3 phosphates are there, out of this 2 phosphates will be removed, and now it will be called as deoxyribonucleotide. When it breaks the 2 phosphate bonds, it will get the energy. The energy is released. That energy is used to link up this deoxyribonucleotides opposite to the template strands of the DNA. So, in that way, this deoxyribonucleoside triphosphates will be acting as substrates, as well as they provide the energy for the polymerization. So, they serve dual purposes. And based on the template sequence, that particular deoxyribonucleotide will be joined here, and on the template of 3 prime to 5 prime, the strand synthesis will be happening continuously towards the fork end, or towards the replication end, towards the Y-shaped replication fork. On the opposite strand with a polarity of 5 prime to 3 prime, okay, you have the first one 5 prime to 3 prime. So, in this way what happens is, here primer got added. After primer got added, the deoxyribonucleotides are added in the opposite direction on the template in this way. And once it is done, this part of the DNA got opened now. This much stretch of the DNA got opened. Again, for this strand there is no problem, because it is the direction of replication and the direction of the new strand synthesis are happening in the same direction. So, this strand will be formed continuously on the template of 3 prime to 5 prime, because the opening of the DNA and the replication of DNA is happening in the same direction. But when it comes to the down strand with a polarity of 5 prime to 3 prime, here what happens is, the strand of 5 prime to 3 prime, here the replication is happening towards this end, but the direction of opening is happening in this way, and the replication is happening in this way. it is happening in the opposite direction. So, opening is like this and replication is in this way. So, first RNA primer, it is added here and DNA polymerase polymerized up to this end. Then this much stretch is got opened. Next, one more RNA primer is required. This is the RNA primer, one more red one RNA primer that got added at the replication fork, and now DNA polymerase will be polymerizing in the 5' to 3' direction, because this primer will be acting as the 5' end. So, it will be directing up to the 3' end, the strand synthesis. So, in this way, as the replication and opening are happening in the reverse directions or in the opposite directions on the polarity strand of 5' to 3', always the new DNA strand is formed like fragments, and these fragments are called Okazaki fragments. This strand is formed actually discontinuously, but this strand is formed continuously. So, what you have to remember here is always template of 3' to 5' polarity forms continuous strand. The template which is having the 3' to 5' polarity will always form the continuous strand, because it will be starting with the 5' and the direction of replication and the opening of the DNA is in the same direction. Opening and replication in same direction. But the template of 5' to 3' polarity will form discontinuous strand because opening and replication are happening in the reverse direction. In opposite direction. So, this point you have to remember, and whatever the strand that is synthesized continuously, it is called as, this continuous strand is called as leading strand. And this strand, it is called as, discontinuous strand is called as lagging strand. Okay. So, you can see here very clearly the template of polarity, 3' to 5'. So, this is opening of the DNA and replication of DNA is happening in the same direction. You can see this is replication, this is the replication and this is the opening of the DNA. So, both are happening in the same direction. So, on the template of 3' to 5', continuous strand will be synthesized and this is called as leading strand. And whereas the template of 5' to 3', here RNA primers will be added and the strands, every time one RNA primer is required, because the synthesis of new strand is happening away from the fork end and the direction of opening is in the opposite direction. So, this strand requires many RNA primers. How much ever the DNA is opening, that many RNA primers are required here, and here only one RNA primer is required here, and it is happening always, the synthesis of new strand is happening towards the fork end and here it is going away from the fork end. So, many RNA primers are required because the replication is in this direction, opening is in this direction. So, this is discontinuous synthesized strand. These are called as Okazaki fragments. And this Okazaki fragment, for every Okazaki fragment we need one RNA primer here, and in this side only one RNA primer is enough because both are happening in the same direction. So now what happens, this discontinuous strand which is synthesized in the form of Okazaki fragments, here gaps are there. This RNA primers will be removed by the DNA polymerase, one type will be there, DNA polymerase 1. It will remove that RNA primer. And then DNA ligase enzyme is there. DNA ligase. This DNA ligase will synthesize the phosphodiester bonds between these two. Phosphodiester bonds will be synthesized like this. And now this strand, discontinuously synthesized strand will be there, that is called as a lagging strand. So in this way, after the replication, we will have the two daughter DNAs where one is a parental strand, other one is a newly synthesized strand. So in this way, the DNA replication will be completed until the end. And this requires very much energy. So high energy requirement is there and it is an energy expensive process. So that energy is actually provided by that deoxyribonucleoside triphosphates, or apart from that also sometimes ATP is also used, but generally the deoxyribonucleoside triphosphate, they serve for dual purposes. They act as substrates and they also provide energy for the polymerization. In this way the replication will be happening. This is the diagram which is given in your textbook. So you have to follow this one. Remember, the template of the polarity 3' to 5' always synthesizes the continuous strand which is called as leading strand, because the DNA opening and the replication is in the same direction. On the template of 5' to 3', on this template 5' to 3', the newly synthesized DNA strand is always formed in the discontinuous manner because the replication is happening in this direction and the opening is happening in the opposite direction. And this discontinuous strand is called as lagging strand, which is formed by the joining of Okazaki fragments. This is a very important NEET topic for the upcoming NEET or always, because it is the first step which is going to happen in the S phase of the interphase of cell cycle. Imagine if DNA replication is happening but cell cycle is not completed, then the cells will become polyploid, which is one kind of chromosomal anomaly. And you know, a point where the DNA replication starts, that is called as ORI, origin of replication. So if you want to multiply any particular fragment of DNA, definitely that should have one ORI. ORI site should be there, origin of replication site should be there. In rDNA technology, generally for a DNA fragment, we link up the ORI site so that we can multiply, we can start the replication. And you should remember that in eukaryotic DNA, we have so many ORI sites. But in the prokaryotic DNA, we will have generally only one ORI site will be there. And here, many ORI sites will be there and it will open up like a nodule, the DNA. At one point, it will open and it will proceed to both the ends. And then the replication will be proceeding. So in this way, the replication will be completed. For this, we have to remember one Michelson and Stahl experiment with the problems they will give. They will ask how many strands are heavy, how many strands are like light DNAs, how many are hybrids, how much percentage, that is important there. And here, which polarity will be having the continuous strand, which polarity will be having the discontinuous strand. You can see here already I have written the 3' to 5' is continuous leading strand and 5' to 3' template strand will be forming the lagging strand. So with this, we have completed the DNA replication. Thank you.